Thanks very much. This is work that was done by myself and Klaus's student, Justyne Horodyska, who can't be here today. This is following on from the experiments done in Moore Park, and the animals are divergent for residual feed intake. 20 max growth gilts were selected, 10 high and 10 low RFI. Muscle was taken as close to slaughter as possible and preserved in RNA-later for downstream transcriptomic analysis. Justyne then transported the samples to Germany, and she carried out this work in FPN while I was otherwise involved with small babies. And she extracted the RNA, processed it and hybridized it to the Affymetrix snowball array. Following quality control, mixed model analysis and pathway analysis, the results are that approximately 400 genes were differentially expressed at least 1.5-fold. So there was at least a 1.5-fold either increase or decrease in gene expression in relation to the trait. The most upregulated gene was AP2M1, which is involved in molecular transport. The most downregulated gene was NCOA2, which is an inhibitor of growth. So this is in terms of more efficient animals. In order to try and put some shape on the 400 gene list, the list of 400 genes, we carried out pathway analysis in Ingenuity, and we can see the most significant molecular and cellular functions that were modulated by RFI, a network of genes interacting and pathways. Now you can have a look at the poster yourself when you have a chance, but many of the functions related to accumulation of fatty acid, differentiating between saturated and fatty acid and phospholipids. There was also, so we only show the very top most significant functions here, but there was a long list, and many included connective tissue adhesion and apoptosis. And this was reflected then when you go down to look at individual canonical pathways. So these are known pathways which a significant number of genes were represented within our differentially expressed gene data set. And the most significant signaling pathway was thyroid hormone retinoic acid receptor activation, which is involved again in growth, inhibition, PEDF signaling, which is inhibition of proteolysis, HIF1 alpha signaling, which is apoptosis, and then a number of adipogenesis pathways as well. So these are modulated. So the next step for Justyne will be to validate this array experiment using real-time PCR. And we did note that many of these functions are also important in meat quality, and many of the genes that we saw have previously been shown to be associated with meat quality. So we'll be very interested to look at the meat quality in this model as well.